In the last few lectures, we've learned a lot about the different factors that determine the structure of our proteins, right? We looked at how interactions occur between the peptide bonds of our amino acids, as well as between the side chains of our amino acids in order to stabilize different structures. But we really haven't discussed the process of forming that structure, right? We talked about what happens, but we don't talk about how it happens. So this lecture is going to focus on that process itself. This is called protein folding, right? It's how we go from a linear amino acid chain to that complex 3D structure. So how are folded proteins made? Now, of course, our protein synthesis process starts with the ribosome, right? So the ribosome uses our mRNA to synthesize our peptide sequence based on the RNA sequence, which of course came from our DNA sequence. Now, of course, the initial product is just a chain of amino acids. Now, as soon as that chain of amino acids is produced, it's going to start to fold, right? So it's forming those interactions between our amino acids. So you'll start to see secondary structures popping up, and we'll get these folding intermediates where you have part of your protein folded, but maybe it's not all the way folded and hasn't arrived at that final folded native structure. After some time of folding, it will then arrive at our native structure, shown over here. And of course, then that protein, once it forms that particular structure, it will be able to carry out its specific function. Once the protein is no longer needed, it will eventually be degraded then into peptide fragments as we complete the life cycle of our protein. Now, this is, of course, a bit of an oversimplification of the full life cycle of our protein, right? So, of course, the folding process itself, we kind of passed over a bit here. This process is quite complex, and we'll have proteins we call chaperones that will actually help with this process. Our protein can also be unfolded. This is a process we call denaturation. There are also several other potential outcomes for our protein, right? So as our protein is folded, there are a number of different pitfalls that we can have, and we can have our protein aggregate. So this is when multiple semi or unfolded proteins combine together to make larger structures. So this is kind of like the quaternary structure that we talked about, but it's less defined structure and more of just kind of proteins clumping together without a specific structural feature to it. So we get these kinds of aggregates where proteins just kind of crowding on top of each other, and these will generally be degraded again, as with our proteins at the end of their life cycle. We can also have proteins that can misfold, right? So you can end up with proteins not folding correctly in the normal native structure, which can again lead to potential aggregation, or they can be degraded because, of course, if they aren't in that correct structure, they're not going to be doing their correct function. So as we look at the folding process for a particular protein, you can imagine that there might be many possible paths for folding one protein. So here we have an example. We've got our amino acid sequence here, and we want to try to fold this up into a protein. Now you might have one segment that's going to fold into a beta sheet. So this early segment here, another segment that's going to form into an alpha helix, and a third segment that's going to form into another beta sheet. Now, which order do we fold this, right? There are many different possible paths, right? You could start off at the first one, kind of form that beta sheet, and then form the second beta sheet on top of it. Or you could form the first beta sheet and then the alpha helix and start to make like a beta alpha beta loop. There are many different ways that you could approach this folding process. So for any given protein, right, you can imagine hundreds or even thousands or millions of different possible ways to try to fold that protein, just based on the different combinations of what's folding to make what secondary structures, what folds first, which secondary structures are interacting with each other to form that tertiary structure. So it's really quite complex to kind of find that one true structure, right? There are so many different possible paths you can take and different possible outcomes based on the path you take. One of the ways that we can illustrate that is with this free energy funnel diagrams. So for most proteins, our native structure is the lowest possible free energy for that protein, right? Our native structure is the most stable structure, which of course would also have the lowest free energy. So you can represent this with our N down here, and this is at the bottom of our free energy funnel, right? So in our funnel, low free energy is at the bottom, high free energy is up here at the top. And so we're always moving from our extended unfolded structure down to this most stable native folded structure. So over here on the left, we see a funnel representing the fact that for many proteins, we'll have multiple different pathways that can all lead to one stable outcome, right? So on the previous slide, we saw, okay, we could do beta sheet, alpha helix, beta sheet, or we could do beta sheet, alpha helix, and still get to that same structure in both cases, right? So we could come from this direction doing the beta sheet first, and we can still fall down that free energy gradient to reach our native folded structure. Or we could start from this end and form the alpha helix first, and we'll still follow our free energy gradient down to our native folded structure. So this is kind of the ideal situation for a protein, right? We want to have, we're always going to have multiple different paths, and we want them all to kind of flow fluidly down to that native folded structure. For many of our proteins, we'll end up with stable intermediates on the way. So to represent this, we would need something more like our second funnel here. We see all these bumps along the path. So these represent different structures that might have be more unstable as we're trying to get to that native folded structure. So if we are trying to fold this protein here, it's actually going to be highly dependent on where we start, right? So if we fold it from over here, as we're coming down, we're going to be hitting all these higher energy intermediates in the way. And so we can actually get stuck at these half-folded intermediates here because it's more stable than the surrounding intermediates, even though it's less stable than that native folded structure. So this is where we get a lot of the issues, potentially misfolding proteins. So it could be a fold that's not as good as the native fold, but it's better than the other possibilities around it. So as we'll see, our chaperones will be available to try to help deal with these sorts of scenarios where we're stuck in that kind of misfolded structure. We also have another couple options represented over here on the right. So you could have just kind of one stable outcome with a lot of different intermediate folding outcomes at higher free energy. So you have a very sharp funnel here. Or you can also have a significantly stable intermediate kind of from all different perspectives that's kind of blocking your way to the final folded product. And of course, you can imagine other different diagrams as well. These are just kind of showing some of the different potential folding paths that you might see for a particular protein. Now, it's important to note, as we're looking at these free energy funnels, that our proteins are not just kind of randomly trying out different conformations, right? It's actually mathematically impossible for a protein to just randomly rearrange itself until it finds that particular outcome, right? So there's so many different possible combinations of how you can fold a protein when you have hundreds of amino acids that you can't try every possible folding until you find the right one. So we're always kind of moving along that gradient, again, following the path of our funnel to try to find our native structure, right? The native structure is what it is because it's thermodynamically most favorable. It's a structure where we get the best interactions between our amino acid side chains and all that we looked at in the previous lectures. So we can't just explore any possible option we want as we're seeking to get that native structure. We're always kind of following that gradient. And if there's a bump that's blocking us from proceeding, that's going to affect our folding. So we're going to need some way to get around any obstacles that we find in the folding process. So this is accomplished through chaperones. So again, chaperones are proteins, but they're proteins that help other proteins fold. So chaperones, of course, have many different roles. The first role, of course, is fairly obvious. They help proteins fold. They do this by helping proteins to get past those stable intermediates. So they'll often be spending energy in order to help push a protein past kind of one of those free energy barriers in order to get them down to that native folded structure. So this is normally going to require energy. They're going to be binding to the protein in some way, and then helping it to rearrange itself. Now, as it helps the protein to fold, so it can actually do this in a couple different ways. One is directly by spending energy, right? So it might be using ATP to help speed up this process, but it can also help just by restricting the conformation of our protein, right? So just by binding onto our protein, it's kind of restricted the protein, so it can't move around as much, and it can only explore certain potential folded structures, right? So just the binding itself can help to help the protein to fold into that correct structure. So it restricts conformations, and it can also directly cost ATP. Now, another key role for our chaperones is also preventing aggregation of our unfolded peptides. So again, I mentioned before that our proteins can also just clump together and form these sort of aggregates where proteins are just kind of randomly associating together. This often occurs through kind of hydrophobic patches of our proteins just sticking together before they're able to fold and get those hydrophobic patches hidden on the inside of our globular proteins. So if we bind to our protein in our chaperone, then we can kind of keep our protein away from other unfolded or semi-folded proteins, and we can prevent that aggregation. So we prevent aggregation. Again, we can accomplish this by binding to the protein and prevent binding to other folding intermediates. So of course, in this diagram, we see a specific chaperone, this HSP70, binding to our folding intermediate here. And then we see a change in our conformation of our chaperone as it's helping to fold the protein, and it's using ATP to accomplish this. To help illustrate the functions of chaperones, I want to take a closer look at a chaperone system found in E. coli. So this chaperone system uses a protein complex with this GRO-ES protein complex and a GRO-EL protein complex. So here's a diagram of the folding process with this GRO-EL, GRO-ES chaperone. So our GRO-EL and GRO-ES both form a heptamer. So you can see these individual kind of ovals here are representing individual subunits of our GRO-EL. You can also see that the GRO-ES kind of matches that. These will heterodimerize together, and then they also dimerize with another unit of our GRO-EL and GRO-ES system. So you can see that down here in the lower half in the red and the orange, and we have our gray and the orange up here in the upper half. So the first thing that we're going to see is we have a folding intermediate, which is actually delivered by another chaperone to our GRO-EL system. So this is going to bind into our kind of barrel here with our heptamer surrounding it. And it's going to bind here using hydrophobic interaction between, there's some hydrophobic exposed residues within this chamber here, to, of course, expose hydrophobic residues on our unfolded or semi-folded protein. So we bind within this kind of hydrophobic binding pocket inside of our chaperone. And then we also bind 7-ATP on here. and this will trigger the association of our GrowES on top here to now restrict our protein within our chaperone. So at this point, we have our protein inside, and the protein, of course, is going to be restricted because it's bound inside of this structure here. So we already see our first kind of goal. We restrict the conformations of our protein in order to help improve the folding process, right? So if we kind of restrict the movement of the protein, we limit the number of different conformations that that protein can take, right? It can't just randomly flop around, and that will help it to find that native folded structure. Now, the next thing that we're going to do is we're going to use some energy expenditure to help our folding process. So we're going to hydrolyze our ATP. So all 7 ATP, 1 per subunit, are hydrolyzed. And this provides energy to create a conformational change within our structure. We have a surface that changes from hydrophobic to hydrophilic, and that helps to push our protein to fold and hide its hydrophobic residues inside instead of binding just to our protein pocket there. So change in conformation, and that forces our hydrophobic amino acids inside. So with that added energy and, of course, the restricted conformation, our protein is now able to fully fold, and we get our native protein structure. Now, at this point, we just have to kick our protein out. And this is where the dimer comes in with our second GrowEL and GrowES unit down here on the bottom in red. As a new folding intermediate comes in and binds to our empty side of our dimer, and our ATP binds as well, this actually kicks off our GrowES cap on the other end and releases our native protein. Or in some cases, this will just, again, release another slow folding intermediate that will complete its folding on its own after we've kind of helped it along. So you can see we kind of have both sides of our dimer system working in tandem, right? We now have our second half that's working on folding as we move back towards our original structure, and then our new unfolded intermediate comes in and kicks out that folded structure, and we continue this cycle. Right, so one full cycle results in 2 proteins getting folded as each half of our chaperone is kind of working together here. So again, notice the key factors that are at play here with this chaperone. We are restricting the conformations of our protein. Obviously, we've restricted it from aggregating with other proteins because it's stuck by itself inside of our little barrel here. We help to force a change in conformation by spending energy and changing the conformation of our chaperone, and then we're able to release our folded structure. Once we get to our folded structure, of course, then our protein is free to go out and carry out its specific function, which is dependent on that native structure. Now, I mentioned before, of course, that this folding is not irreversible, right? Once we fold to our native protein structure, we can still potentially lose that structure and return to an unfolded structure. This occurs through a process we call denaturation. Now, since, of course, structure is always linked to our function, denaturation also eliminates the function of the protein. So it's a great danger for our proteins, right? If you become denatured, the protein is no longer functional, and you just need to get rid of that protein and get a new one. So what can cause our protein to unfold? Well, there are actually a lot of different factors. Heat and cold can both cause denaturation, extremes of pH, organic solvents, as well as some different what we call chaotropic agents, are urea and guanidinium chloride, for example. Now, if we're going to denature a protein, essentially what we need to do is we need to disrupt those key interactions that help our protein to fold and form that specific native structure. So each of our different denaturing agents will be targeting different interactions within our protein. We'll disrupt those interactions in order to help our protein to unfold. So with our first example here, we're going to look at how we can interrupt a electrostatic interaction within our protein, right? So our protein relies on interactions between positive and negatively charged amino acids to help hold together our tertiary structure. Now, when we add guanidinium chloride, so guanidinium chloride is this ionic compound here with a positive charge and a negative charge. Now, if we have very high concentrations of this, it will interact with our charged amino acids, and it can actually disrupt and break up the normal interactions that we would have with our positively charged amino acids, right? So notice we've kind of replaced our arginine here with our guanidinium, and now our guanidinium is pairing up with our negatively charged amino acids, our aspartates and our glutamates, instead of our arginine, right? You can see each of our negatively charged amino acids is now paired up with that guanidinium. So essentially it's just competing here, and it's shielding and blocking out the normal interactions, the normal salt bridges that we would form to help hold our protein together. Changing the temperature can also have a significant effect on our protein structure. Now, of course, this is a denaturation mechanism that we're very familiar with because we use this very commonly in food preparation, right? When you cook an egg, you're actually denaturing the proteins of the egg, and then they're clumping together to form the solid cooked egg that we then go on to eat. So that solid structure of our egg is actually formed because of proteins unfolding and then aggregating together to create this more. So how does heat actually break up our protein structure? Well, it does it by increasing the conformational flexibility of our protein, right? So one thing that we learn about in Gen Chem is that heat, of course, increases the kinetic energy of our atoms and our molecules, right? So as you add that heat, our molecules and our atoms start jiggling around more. They're trying to explore different conformations. Within our protein, they're all restricted into a very tight conformation. As they gain more energy and they keep moving around, they will eventually break out of those interactions that are holding them in that specific structure, and then that results in our protein unfolding, right? So we get this kind of thermal unfolding process that's shown here. This will usually start with our most free areas of the protein. So it starts kind of down here where you've got a little bit more freedom in general. It starts to pull apart as those interactions are broken because of the energy that we have here. And then, of course, that kind of cascades throughout our whole protein. It just starts to fall apart as we are exploring different conformational options here. So our proteins can essentially be considered to have a melting temperature, right? At some point, as we increase the temperature, we increase that energy, and then our protein kind of falls apart. Now, of course, it's a little bit different than a true kind of solid to liquid transition. It's actually a folded to unfolded transition, but it behaves kind of similarly to a normal melting point. So in this graph here, we can see different melting points for different proteins. And we have our percent unfolded on the y-axis. So at 0 down here, this would be our folded native protein. And then at the top, we have our fully denatured protein at 100% unfolded. And you can see each protein will have a specific melting point where once we hit that temperature, our protein unfolds and becomes denatured. That melting point will be different between different proteins, right? So notice we have ribonuclease over here on the left with the melting point around 50-some degrees, whereas this apomyoglobin has a higher melting point above 60 degrees. This is something that we commonly see, right? Some proteins will be more able to resist those changes in temperature. Others will be a lot more vulnerable to temperature changes. Now, the main key for this is just that you need a protein that has stronger forces holding the protein together if you want that protein to survive in hot environments, right? So there are bacteria, for example, that live in hot springs. And so they have very heat-resistant proteins, and they're just going to have much stronger tertiary interactions within that tertiary structure in order to keep those proteins together as that heat increases, right? So you might see a lot of salt bridges, for example, to hold that together. Now, the same sort of denaturation graph can also be used for other denaturation methods. So notice over here we have a second graph looking at the addition of guanidinium chloride. So as we increase the concentration, you can see again we hit kind of a transition point where we move quickly now from our folded state to our unfolded state. So the graph really looks quite similar. It's just whether you're using heat to denature it or whether you're using higher concentrations of your chaotropic agent here. Now, I should note, of course, it does require very high concentrations, right? Because again, you're essentially competing against the interactions between your native amino acids when you're adding in this sort of agent here. The next denaturation agent that I want to look at is beta-mercaptoethanol. You can see it down here in the lower left corner. So, of course, one of the things that helps to hold our proteins together is disulfide bonds. Disulfide bonds, of course, are a very important part of our tertiary structure because they allow the formation of covalent bonds between cysteines that are within our amino acid sequence, right? So remember, two cysteines can bond together in an oxidation reaction to create this disulfide linkage between those two amino acids. However, when we have molecules like our beta-mercaptoethanol, we can also have a reduction reaction that can break apart these disulfide bonds to restore our original cysteines. So any kind of reducing agent like beta-mercaptoethanol can disrupt those disulfide bonds and then help our protein to fall apart. The Anfinsen experiment was one of the seminal papers that helped to show this sort of denaturation process. So this was actually awarded the Nobel Prize in 1972. So in the experiment, he took the ribonuclease protein with its native folded structure, and you can see there are several key disulfide bonds marked in yellow within the tertiary structure of the protein. So he added beta-mercaptoethanol to reduce those disulfide bonds. He also added urea. So urea kind of helps to override some of the hydrogen bonding and break up the structure as well, kind of similar to guanidinium chloride. So once he added those denaturing agents, he was able to break up the interactions and form an unfolded structure for the protein here. Now, once you removed the urea and the mercaptoethanol, the structure actually refolded into the native state. So depending on the protein, some proteins are able to reversibly denature and then fold themselves again into the correct structure. However, there are many proteins that are unable to do this, right? Once they're denatured, they either aggregate or they're just unable to find that native structure again without the help of chaperones. So again, if our protein is denatured and is not able to refold, we have the possibility of forming aggregates. So you can notice here in this diagram, so we have our native structure. It can go to the unfolded structure if we add various denaturing agents. And it can go to the folded structure, again, depending on how easy it is for it to find that correct conformation. But if it finds other unfolded proteins, right, once our protein is unfolded, we're going to have kind of our hydrophobic faces of our protein exposed, right? Normally they're all hidden inside as we form that compact globular protein structure. But if we're unfolded, we no longer have that hidden. So these hydrophobic faces, of course, want to clump together following our hydrophobic effect. So this protein might grab onto the hydrophobic portion of that protein, and we can then end up clumping together. Right, so this is a negative consequence of the hydrophobic effect, right? Any unfolded protein sitting around will really want to clump together so that we can increase our entropy, right? Again, releasing that ordered water that we have coating any hydrophobic surface within our cells. These sorts of aggregates can have many different health consequences, right? The most obvious consequence is that we lose the original function of that protein, right? So the protein is no longer functional and we have to produce a new protein. Generally, this isn't a huge problem for our cells because we are continually producing new proteins, but it can be inconvenient. However, the other problem that we have is as we are aggregating, we can also create kind of large structures that can interfere with our cell's normal function, right? So the beta amyloid protein is known to self-associate in kind of a partially unfolded or misfolded state. So it's not a true kind of denatured aggregate, but it is kind of a misfolded aggregate. And it can actually combine together using multiple units to form, you can see this kind of extended beta sheet as we're packing multiple amyloid beta units together. And you can form this amyloid fibril. It's been proposed that these amyloid fibrils are actually part of what causes things like Alzheimer's disease. So these end up expanding into larger filaments as we interact with multiple beta amyloid proteins and kind of stack them on top of each other. So this is kind of an interesting case where it's not a true denatured aggregate of just kind of different proteins clumping together, but it's a more ordered aggregate of our amyloid beta proteins. We also see this sort of protein misfolding in other diseases as well. So if you've heard of prion-based diseases, such as mad cow disease, these actually use unfolded proteins as kind of infectious disease proteins. So the thought is that you get this prion protein that's kind of misfolded in a way, and then it aggregates with other proteins that actually encourages other proteins to misfold as well and aggregate together with the prion. So it's not infectious in the form of a virus, but it's infectious in the form of an unfolded protein that helps other proteins to unfold, kind of like the opposite of a chaperone. So as we can see here, the protein folding and unfolding process is quite complex, right? We have different factors and we have chaperones that will help us to fold, but then there are also many different factors that can help us to unfold. And again, as we see, this is very important for our biochemistry and our health in general, as that folding is critical for the correct function of our proteins.